Yeah, thanks for the intro, and thanks to everyone for coming here this afternoon to listen to my talk, and thanks to the BQI25 team for inviting me here today. So yeah, some of this is already covered in terms of my background. Yeah, so within the Centre for Crop and Disease Management at Curtin Uni, I'm in the fungicide resistance group, so basically a group within the centre, and we're all, me and my colleagues are all doing fungicide resistance, obviously, and yeah, most of my colleagues are in cereals, and I'm the Lone Ranger doing grapes, basically, in the group. So yeah, currently involved in a CRC SAFE funded project looking at fungicide resistance in 3 pathogens in grapevines, so that's your Botrytis, cinerea, and your 2 mildews. So that's not what I'm going to talk about today. So basically, a couple of years ago, we ran a Curtin, a master's project at Curtin University with a student. So we took our skills with botrytis and grapes, and we looked at basically botrytis or fungicide resistance and botrytis from small fruit and strawberry runners. So that's what we're going to talk about today. So that was Namala Sharma, the student. So Fran Lopez, my line manager, and myself, we managed Namala. So Namala's left the centre, so otherwise she would have been here today because she did all the grunt work. Okay, so just the project outline. So it's a these masters projects at Curtin, they're very short. They're only about 6 months, so we don't have a lot of time. So basically, kept it really simple. So basically, in 2021 and 2022, we acquired fruit from retailers. So that was strawberries, mostly strawberries, blackberries, blueberries, and raspberries. We also got hold of some strawberry runners. Basically, yeah, very simple stuff here, simple bucket science. We just incubate the fruit and the runners in plastic trays with cling film over top and moist, basically tissue paper. Incubate them at room temperature, and you get a whole bunch of stuff growing, as you can see in the photo. There wasn't a lot of sporulation, but we eventually worked out picking out a certain type of fluffy white stuff was botrytis. There's some other stuff in there, but a fair amount of botrytis. So from there we basically extract it and single spore. So we'd like to produce pure strains. So that's how, in the lab, we want to work with a pure strain in terms of the screening on fungicides and the genetic stuff. So from there, yeah, we basically create a population of pure strains, and then we screen the isolates on a bunch of different doses. So just one dose per fungicide, so it makes it very, very quick and simple. So a lot of those doses came out of my work with grapes in terms of basically one dose to tell you if it's resistant or not. And ultimately, we do a bit of genetic work, so we want to associate a mutation, ideally, with that resistance that we found. So just a bit of a explainer around how fungicide resistance develops. So basically, you got a crop. You apply fungicide. So already in that crop, there will be naturally occurring resistant strains present in the population. So they're obviously at very low frequency. So there's a whole bunch of mutants sitting out there, but there's been no selection, so they're at very low frequency. So with that first application, you're basically getting essentially full control because the resistant isolates are at very low levels. But obviously over time, with further applications throughout seasons, you get an increase in frequency of these resistant survivors or resistant strains, whatever you want to call it. Okay, so yeah, we got around about 300 isolates from this project. So it's a fair chunk of isolates, which is really nice for Namala, considering the projects aren't that long. Most of these isolates are from strawberry under conventional management. So basically the graph shows basically 3/4 were strawberry from conventional management. The numbers there indicate the number of sources. So we had 13 sources too for the strawberry conventional samples, which is really nice. So all the other types, you can see there's only one or two sources, so not a lot, and not many isolates too. So 2 types of blueberry, and then the blackberry and the raspberry and the strawberry runners. So the fruit, all appeared to be from WA. And the runners originated from Victoria and Queensland. Screening on fungicides, as I mentioned earlier, it's very simple, just one dose, all these different strains on one dose of fungicide, so basically a yes or no answer. The table is just showing all the chemistry we tested, so you've got groups 1, 2, 3, 7, and 9, 11, 12, and 17. So they're all registered in strawberries and blueberries, except group 1, which used to be registered, so it's been deregistered. So that was just curiosity thing really. So we could just, while you're doing the screening, you usually do as much as you can because it's fairly easy to do it all at once. So that was just interesting to do the group 1 as well. So the results. So this is just for strawberry. So I've broken it into the strawberry conventional isolates, strawberry organic, and the runners. So I'll just explain the first figure. So basically on the horizontal axis, you've got all the different chemical groups that we tested against, and obviously sensitive slop. On the vertical axis, we've got the frequency. So basically the number of isolates that were resistant to each of those chemicals. So these graphs, they don't consider multi-resistance. So there could be isolates that sit across those different columns because they're resistant to multiple fungicides. So they can sit anywhere within there. So it's just purely on a one fungicide as you go along the graph. So they're not, the bars aren't going to add up to 100%. So that's why some of these bars are really high and clustered to the right. So with the first graph, it was the conventional strawberry samples. It was really nice because we had 200 isolates. So that was a decent chunk. If you look at the other two, there's only 26 or 27 isolates, which isn't a lot to work with. I think it's nice having a bit more to make more what do you call it? More confident conclusions and speculation. Yes. So if you look at the first graph, so frequencies are fairly high on that right side of the graph. So your group 2, 17, 9, and 11. So your group 2 and group 17 and group 9, they're classic botryticides, and they've been around a long time, and they've been heavily relied upon, and that's probably really showing in the data. Group 11 is interesting because I know in grapes it's not heavily used to control botrytis. I'm not sure about in small fruit. So besides there being selection here, or being used as a botryticide, and it's selected for resistance, some of this resistance in botrytis might be due to off-target selection. So basically you've used the group 11 to control powdery mildew, and when you've applied that to control mildew, you've actually selected for resistance in botrytis. So we call that off-target selection. For the other groups, you've got group 3, the DMIs. So that's like group 11. It's not that effective on botrytis. So looking at this graph, to me, I'm assuming it hasn't been used that much because it's not very effective on botrytis, and the frequencies are very low. And botrytis doesn't seem to get resistant to group 3 as well. So it's very rare to find that in the literature that it's resistant to that group. Group 12. So generally with group 12, it's always used in combination. That's my understanding. I think it's always used with another chemical group. And also, when it gets resistant, it generally has a very high fitness cost. So with a high fitness cost, it will struggle to grow and survive in the wild. So you might get it might develop and become a resistant isolate initially, but it will struggle to outcompete any other isolates. So probably over time, it just gets outcompeted. So that's why you see the frequencies so low. Group 1 was the deregistered one I mentioned earlier, carbenism. So that's fairly low, and you might wonder why that's still there. But there's a thing called selection by association. So basically, in the past, a number of isolates might have become resistant to group 1. We've gone on to other chemistry and maintained group 1 by using other chemistry. So you might have a double or a triple mutant, but you can maintain other forms of resistance by just using one chemistry because you've got a double or triple or quadruple or whatever it may be. So that's just an example, just sort of showing you what's the explanation behind that. Group 7, group 7 wasn't as bad as the others, but it's creeping up there. So the strawberry organic one, that was surprising, I guess, having some resistant isolates in the strawberry organic samples. And it looked a little bit like the strawberry conventional, but obviously not as extreme. The runners was fairly clean, except with the group 11, but that's going back to the selection by association again. So I think they're trying to control mildew in runners with the group 11, but in doing that, you're selecting for resistant botrytis. Okay, so I've got a few isolates of blueberry and conventional blueberry and organic blueberry. So not many, so 18 and 14 isolates. So it's not a lot to run with. The conventional blueberry looked a little bit similar to the strawberry graph, but not as extreme. And the organic blueberry was essentially looks like about 85% was sensitive to that giant bar, which kind of makes sense being organic. A few isolates for blackberry and raspberry as well. So again, only 9 or 11, so not a lot, and just one source. These graphs, they did look again quite similar to the strawberry ones. So obviously similar chemistry that's been applied, and similar, So basically very very similar to the what's happening in the strawberry, but with the low sample size, it's probably very hard to be confident that you've obviously you haven't got a large population to look at. So it's not, I wouldn't make the same conclusions as with compared to the strawberry data. So multi-resistance, I touched on that before. Basically, yeah, you've got isolates that are resistant to multiple fungicides simultaneously. So what have we got here? Blue bars, just showing you strawberry. So basically the graph breaks up each isolate into a category where it's resistant to 0 fungicides or right up to 8 fungicides simultaneously. So 0 to 8, and you see it's got a nice sort of, I guess, shape in the middle there. So the majority of isolates are sitting around the 4 to 5 to 6 mark. So most isolates are resistant to that many chemicals at once. We've got the orange bars, so I just combined the blueberry, blackberry and raspberry data together. It looks a little bit similar. There's a few more sensitive isolates, so on the 0. So you might ask why that shape? So I guess there's a number of reasons, number of things that could be happening here. Obviously selection pressure. For things to move around, you obviously need certain types of selection. So if you think things were going to move to the right of the graph, you need selection from other fungicides. Fitness costs, so I mentioned fitness cost comes into play here. So when you're resistant to 7 or 8 fungicides, all those fitness costs, some of them might be small, but they might combine to make you not actually that fit. So that might be another reason why there's not many down the far end, down the right end. And obviously evolution. So this data is now, 3 or 4 years old now. So if we did this all again tomorrow, would it look different? Because it's 3 or 4 years have passed, you know. Okay, genotyping. Not to bog you down with techno-babble. So basically, we looked at a few key groups. So group 2, 7, 9, and 17. They've all at least got one gene that we can look at, and they'll at least have one mutation. So yeah, you can either have one gene for say group 2, or for group 7 you can have 3 genes. And yeah, 2 mutations for group 9, or at least 10 mutations for group 7. So it can get quite complex. So the graph on the right was basically just showing, we took a whole bunch of different genotypes or different mutants for group 7 resistance to boscalid. We did a thing called an EC50, so we tested the isolates on different doses of fungicide as a way to measure their sensitivity more than just one dose, and you create an EC50, which is basically the dose of fungicide that reduces growth by 50%. But all I was just really putting this graph up here is to show you basically how complex the system can be. So you've got a bunch of different genotypes, a bunch of different sensitivities. So in the field, it could be quite complex with a different type of chemistry you're using. So there might be, I'm not sure in strawberry how many different group 7 fungicides are available, but some of the group 7s might have different effects on different genotypes. So that was just an example of how complex it can basically get. It's probably a bit simpler than in some of the other groups, but the group 7s are fairly complex with the 3 genes and all the different changes. So the conclusions, for strawberry, conventional frequencies were fairly high. You know, you're looking at 80 or even more than 80%, which was similar to some of the overseas ones, but actually higher than some of the overseas ones. And some of the overseas ones were earlier than ours. And high frequencies to these groups, so we're talking about on the right-hand side of those graphs, are likely just to reflect the long-term usage and reliance on those chemical groups. We talked about high frequencies to group 11 as potentially just this off-target selection. You're using a group 11 to control the mildew. Low frequencies, so you've got fitness costs that might affect group 12. So you might have low frequency of group 12 due to fitness cost. Low usage, group 3, and then the deregistration, group 1. But then that potential selection by association there. Multi-resistance, we just covered that. Yeah, so obviously lots of things at play there. Fitness costs, the type of pressure, and obviously like the stage and resistance development. So the other fruit, like I've said a couple of times, the sample sizes are very small, very low. So it's hard to probably be as confident with what I'm saying in terms of conclusions compared to the strawberry data. But the frequencies were similar to reports overseas. So managing resistance. So when I look at say strawberry conventional, when you see these crazy frequencies for some of these groups, very high frequencies, you might find some of this chemistry has become less effective. So here at this stage, you'd look at focusing on improving cultural practices and obviously your cultivars. And another thing I was just thinking, I was talking to Stuart Dorr before, some of the multi-sites as well. If the multi-site fungicides are available, so that's still obviously something as part of your toolkit, which is be really handy if these single-site actives aren't as good as they used to be with these high frequencies. The resistance present in organic fruit. Yeah, physical dispersal. I mean, have they moved between different sites physically, via humans. Wind dispersal, yeah, between different farms, neighbouring farms, and previous chemical management. So maybe 5-10 years ago, there might have been chemistry applied at that site. So again, it's just speculation, but I guess a couple of reasons why that may be the case. Future work, yeah, it'd be nice to do a bigger study. So this was, like I said, it was only a small study, nice to do a larger scale screening, kind of similar to what we've done in grapes, long-term sort of multi-seasonal screening nationwide, and look at some of these other groups. So we just talked about some of these other groups at lunchtime. So you've got these new groups that people have been using, the group 19, 21 and 52. So it'd be good to see what's happening in those groups. So this year we're doing another master's project, another student. So we are looking at strawberry powdery mildew now. So we're going to basically do nanopore sequencing, where we just look at no growing of the fungus, because it's really hard to grow mildew and time consuming. It's just purely genetic. So we're just going to look at the target genes and look for mutations that have been found to be associated with resistance. So that's going to kick off very soon. The Botrytis fungicide test. So I've just run this at lunchtime, and I'm going to run it again tomorrow at lunchtime. So just a quick run through on this. I know a few of you guys were there at lunchtime, so probably know what I'm talking about. So this is basically just a mobile qPCR machine. So a very rapid test. The reaction runs for an hour. So basically use this in vineyards in the past, and my colleagues have used it in cereals. So basically you can go on site and do in situ resistance detection. And it's based on a very simple DNA extraction method, where you just hand grind the biomass, and it's very quick and easy and simple, and run it through the machine. And yeah, it's adaptable to other pathogens and different assays. So yeah, like I've done Botrytis, done mildew in grapes, and my colleagues have done mildew and cereals. So there is limits. Reaction number, I mean, it's a small mobile machine, so it doesn't have a lot of reaction space. So the reaction number is about 48. So depending if you replicate, or you run standards, or you run controls, that affects how much you can do. The number of mutation targets. So generally in a reaction, you just look at a sensitive and a mutant for that same gene. So that's 2. You can size later in another one. So there's limits on that. And obviously detection limits. It's a crude system to extract DNA. Like, it's really quick and dirty and really easy, but it's crude. So a lab-based method would probably produce better DNA. But then that's lab-based, and it's more time-consuming, and you need bigger equipment, and etc. So, yeah, that's just some of the limits with that. And that's about it for me. So just some acknowledgements. A big thanks to Namala, the student, for doing all that work, slaving away, and Fran, my line manager, for direction in the project. I'd like to thank the Centre for Crop and Disease Management, headed up by the director, Mark Gibbard, at Curtin University, and also my colleagues at the Fungicide Resistance Group within the centre. Thank you.
Thanks so much, Lincoln. Are there any questions?
Yeah, I'm Chris Story from Kendall Plant Care. In your analysis, do you look at the external issues? Are you ever looking at the internal biochemistry of the leaves? Because if you get the bricks of the leaf, which is what you want, which is the basis for all your enzymes and proteins, I think you get an extra thicker leaf surface, which will be resistant to fungal attachment. Do you ever look at bricks in the plants you're looking at, in the biochemistry?
No, we've never gone to that level. So
That's the whole basis for the plant growing, is the leaf. And the biochemistry, you're looking at external factors. If you look at the internal ones, you might find a better resistance to fungal attachment on the leaf surface, because the leaf has inherent mechanisms for rejecting this sort of disease. Salicylic acid is one thing the leaves produce to resist disease incursion.
what about fruit then?
Sorry?
What about fruit?
Sorry?
Fruit.
Fruit.
What about the berries themselves?
I can't hear, I'm sorry. I'm a little
Oh,
bit-
what about the
Use
berries?
the microphone.
What about the berries? What about the berries then? You're talking about leaves. What about the
I'm
berries?
talking about the leaf.
Yeah, but a lot of my samples are from fruit, so where does that come in? Is it a different scenario to a leaf though? Or?
Yeah, I'm just, you talk about all the fungus, etc.
Yeah.
You said you haven't analysed the actual biochemistry in the plant or the leaf, which is
No.
important. In my research, you know, we can reduce the incidence of fungal growth by improving the Brix level of the leaf.
Sure. Oh yeah.
And with higher Brix level, 50% of the sugar in the leaf goes to the fruit and 50% to the
Okay.
roots. So
Yeah.
in grapes that we're testing, we get the Brix up higher to 20, 22. That means the more sugar goes to the grape and you're picking them a month earlier, for example.
Yeah.
And we have less incidence of mildews.
Okay, that's interesting. Yeah.
That's the relationship we're finding.
Okay. Yeah.
Shut up, session as well.
Yeah. Sorry. Just a question on the group 3. Did you use that as a group 3 on its own, or as a mixture with an 11 like Incognito or Veritas or something? Because Teb, I just wanted you to comment on the likelihood of the activity, because a group 3 not particularly active on Botrytis, whether that was a large part of why there's no resistance, because it's not particularly active, compared to a group 11 and some of the newer chemistries that are very, very targeted, you know, to a single site, which the selection pressure is much higher for those individual mutations.
Yeah,
But
yes,
you know, the danger.
Yeah, one idea with group 3 is it's not that effective, so people aren't using it as a standalone, right? So there's no, the frequencies of it. But the other thing is, like I said, in the literature you never see resistance to group 3, but you see it to group 11, so it's different, you have substitute chemistry.
But I think it would only be registered on Botrytis as a mixture.
Yeah, it is.
Whereas
Yeah.
probably the group 11 is the reason it would be registered as a mixture. Because
Yeah, my understanding was only registered, I thought they're both registered as a duo, but I think it's,
but that's what I'm saying, that TEB on its own, you're probably unlikely to ever use it for Botrytis
Yeah.
on its own, because it's always with group 11,
Yep.
which is going to be lifting.
sure, yeah, exactly. And the group 11 can be a standalone, right? Yeah, so cool. You've nailed it. That's probably why. But the big thing with group 11, I think, is this off-target using group 11 on mildews. That's what we think. I mean, it could be a combination, right? Combination of using it for Botrytis, but using it when you're trying to deal with mildew, I guess. Yeah.
Alright, we're running close to time, we're going to do a follow-up question.
Yeah, just interested in the fact that you've gone and taken berries that got to market. So it might have had 5 sprays of whatever, Fungicide, or should it have 1 or something? And then you're incubating, and say he selected what's left on there for that strain, and then you've gone and bred it up. Aren't you just artificially going to just breed that one, which isn't what would have happened in the field?
I mean,
Like, how relevant is what's happening in his growing crop compared to you incubating what's left after it's all been sprayed? All I'm thinking is the Botrytis that's remaining on the berry might only be the survivors. So then you're getting an artificial population.
when ideally I'd
That's
go
what you're
to that same farm and sample every 2 weeks.
Okay.
As you're cycling through, for sure. It's like at the end of the, well,
So is there a link with the grower season, what you're finding? That's what I'm asking.
well, a lot of these samples are from retailers, so we weren't going back
So
and
you don't know?
we weren't chasing up.
Okay.
Yeah, but looking at the results, I feel like there's a fair amount of stuff being thrown on there for a fair number of years. But it's a good point, you know, what stage am I getting these things at? I
And
mean,
you're not testing for any of those actives in residues, are you?
no, I'm not looking at, no, I'm looking at
The organic
residues.
stuff doesn't get tested.
No, but that might explain what, yeah.
But we don't want to know.
I mean, maybe the organic was mislabeled when we're taking it at face value. We might
Yeah.
have gone to a market and got some samples and, oh, you guys organic. So, yeah, it's just speculation, right? Yeah.
Thanks, Lucas, thanks everyone.